Hey biology students, today we are going to start with a review of the light reactions before moving on and putting them together with the Calvin cycle. So what we're going to start with today is looking at an overall picture, a review of the light reactions as we left off with last week. So in order to review this, I've started out by kind of drawing the thylakoid membrane, and I'm taking a few liberties here to keep it simple enough that you can understand what's happening. This is P680 and P700, which are of course our two reaction centers. And so we have photosystem 1 happening here and photosystem 2, which of course happens first. So we have the sun coming in, and we're going to see our wavelength of light coming in to hit P680. At P680, as you remember, we have split water. So when water splits, remember it breaks into three pieces. Water breaks into oxygen, it breaks into hydrogens, and electrons. And at this point now, the electron is going to be hit with this wavelength of light, and it's going to travel up to its excited state, and it's going to travel down the electron transport chain, moving as it goes to P700. Once it reaches down to P700, another wavelength of light is going to come in and strike P700, and this electron is going to jump up to its excited state, and once again it's going to roll down the electron transport chain until it reaches its electron carrier. Remember, we talked about the electron carriers on Friday. NADP is going to pick up the electron from this electron transport chain, and it's also going to pick up another passenger. So thus far, we have not talked about the chemiosmotic gradient. The chemiosmotic gradient works through a molecule called ATP synthase. And what happens in the chemiosmotic gradient, in chemiosmosis, is that we create a large number of hydrogens on one side of the membrane. And on this case, we're going to create this on the interior portion of the thylakoid membrane. So we've got to ask ourselves a few questions here. First of all, where did all these hydrogens come from? Answer, they're being split from water and they're coming on over. Yeah? Good. So they come from splitting of water and other things that may happen in the cell that may give off hydrogens. This energy from the electron in photosystem 2 helps to create the proton gradient. So this is something we're going to add. I'm going to add it as a note right here. The movement of the electron down the electron transport chain of photosystem 2. The movement of the electron down the electron transport chain of photosystem 2 creates the proton gradient. The second question we need to ask ourselves is, what is a proton gradient? Well, a proton gradient means that we have lots of hydrogen over here, lots of H+, and we don't have any over here. It's so sad. We want more over here. And so all of these protons, they want to come over. They want to come to this side of the membrane. But every time they try to get through, this energy of this electron traveling down this electron transport chain creates a barrier, an energy barrier, so that they cannot come through. So they want to come through, but they can't come through. They're trying, but they keep getting blocked. They might try to come through here, but they're not going to be able to make it. Okay, they keep getting turned away. So now we come back to this important molecule, ATP synthase. And ATP synthase is just a protein that is inside of the membrane. And what it's going to do is ATP synthase is going to allow a number of protons to come through the thylakoid membrane. They're going to travel through the ATP synthase molecule. These protons that get to come over here are going to be so happy. There's such a release of energy that they are able to catalyze the making of ATP. Remember, when ATP is made, it changes from ADP to ATP, adenosine diphosphate to adenosine triphosphate. So now we have our NAD, our electron, and our hydrogen, and all of these things together make NADPH. And as a reminder, NADPH and ATP, now that they've been made in the light reactions, are now going to leave to go be a part of the Calvin cycle. My suggestion to you is that you rewind that whole thing and you watch it again, because it is not an easy thing to get all of these details into our heads the first few times we listen to it. The biggest benefit that you have from distance learning is that you can now pause this and rewind and watch it again. If you were in my class, if we were back at Bishop, you would get to see this once and you'd have to rely on your notes for help from there. But now, under these circumstances, you guys get to watch this again. Okay, let me walk you through it one more time before I erase it. So we get our wavelength of sun coming in, hitting P680, causing the electron that comes from water, yes, the splitting of water, causing our electron to jump up to a higher energy level and create this energy gradient that forces all of the protons across into this thylakoid space. Meanwhile, another wavelength of light comes in, excites this electron. The electron's over here at the end of the electron transport chain. It's ready to jump onto NADP, but before it can, it needs a hydrogen to come through the ATP synthase molecule and attach all of these guys together in order to make NADPH. And as a result of this hydrogen coming through the ATP synthase molecule, ADP gets turned into ATP and we get the two products of the light reactions that are necessary for the Calvin cycle, or the light independent reactions. So I've just in our light reactions and I've moved the products of the light reactions, the ones we care about, NADPH and ATP, and I moved them up here into this corner. They used to be way down here in this corner, right? But we're just going to keep on moving. We're continuing this story as it goes. So we are now moving from the light reactions, or the light-dependent reactions of photosynthesis, on to the light-independent reaction of the Calvin cycle. At this moment, NADPH and ATP are going to be coming over to a cycle already in progress. What happens in the Calvin cycle is that the other reactant, besides water, what else do we need for photosynthesis besides water? Let's look at the equation real quickly. This is the equation for photosynthesis. In the light reactions, we took water and we split it and we turned it into O2. We oxidized water because we made it lose electrons. We took the electrons, we gave them to the light reactions. And the light reactions have transferred them temporarily right now into NADPH. Now, in the light independent reactions, or the Calvin cycle, we're going to take 6 carbon dioxide molecules and we're going to reduce them into sugar. We're going to give more and more electrons to CO2 until it becomes glucose. Where do you think it's going to get those electrons from? Well, you're very smart if you think it's going to get them from NADPH. NADPH is holding on to them right now. It took them from water, it's going to give them to carbon dioxide, so carbon dioxide can become sugar. So that is what's going to be happening right now in the Calvin cycle. So let's see what it looks like. So at the start of the Calvin cycle, we're going to see that a molecule called CO2 combines with another molecule already present inside the chloroplast named Rubisco, or RUBP. RUBP, as a reminder, is already present inside of the thylakoid membrane, and we're going to add carbon dioxide to it. So in the beginning of the Calvin cycle, these two molecules combine. And they form a molecule whose name you do not need to know, but they form something. Its name is 3-phosphoglycerate. And 3-phosphoglycerate, which is a reminder, you don't need to know its name, is going to become a molecule called G3P. In order for 3-phosphoglycerate to become like G3P, it needs to gain energy from ATP. So ATP is going to give its energy to 3-phosphoglycerate in order for 3-phosphoglycerate to become G3P. And also, 3-phosphoglycerate is going to need to get some of those high energy electrons that we made in the light reaction. So those high energy electrons that were collected from water and that traveled through the electron transport chains, those high energy electrons are also given to carbon dioxide in order to turn it into G3P. The G3Ps then, with a little more input of energy, the G3Ps turn back into Rubisco and the cycle can continue. But we've missed a really important part. The really important part is figuring out how many of all of these molecules we have. So I'm going to let you now look at this picture that we have in the slides. So we're starting out with Rubisco, and one thing I need to tell you is that each of these little red circles represents one carbon molecule inside of the molecule. And in the front, we have another number that represents how many of those molecules were needed. This can get a little bit tricky. And it's one of those things where typically you guys would ask me a bunch of questions while we were going over this. But since we are at distance, I'm going to go through it now and I'm going to answer your questions tomorrow. No question is too silly. Please, please bring your questions with you to class tomorrow. We start out with Rubisco. There are 3 of them, each with 5 carbons. Let's count them together. 1, 2, 3, 4, 5. So Rubisco, there are 3 molecules of Rubisco, each with 5 carbons, for a grand total, 3 times 5, of 15 carbons. 3 times a 5-carbon sugar gives us 15 carbons. It's going to interact with carbon dioxide. 3 1-carbon carbon dioxide. 3 times 1 is 3 carbons. So now these 2 items get put together, the Rubiscos and the carbon dioxide, and we end up with 6 3-carbon sugars. 6 3-carbon sugars is 18 carbons, and luckily for us, 15 plus 3 also equals 18. So 2 ways our accounting is working right now to give us 18 carbons in the molecule. G3P is also a 3-carbon sugar. So we make 6 G3P molecules. So this is a total of 18. But we're going to let one G3P molecule leaves. That means that we are minus 3 carbons. This one down here has 3 carbons. We're going to come back to that guy in a second, but for now we're going to finish our cycle. So when we get rid of 1, this becomes 15 because that becomes 5 and that becomes then 15. And these get recycled back and we're back to our 15 carbons of Rubisco. So our cycle has turned 1 time around and it has made 1 G3P. Now here's the big crazy thing that needs to happen. In order to make a glucose, we need 2 G3P molecules. In order to make a glucose, I need to do this whole thing again. This whole thing needs to happen times 2 so that I get 2 G3Ps and they can become 1 molecule. So it takes 2 turns of the Calvin cycle in order to make a glucose. It takes 2 total turns of this entire process in order to make just 1 single sugar. It's complicated. So let's read through the instructions as they are said here and let's make sure that you're following. So Rubisco combines with 3 CO2 molecules. ATP energy is consumed, that's right here, and the complex is reduced. So they gain, G3P gains electrons, GR gains electrons to become G3P. 6 G3P are produced. 1 G3P is released to go form glucose, and 5 G3Ps get recycled back to Rubisco to turn the cycle again. After another turn, 2 G3P molecules have been released and they combine to form 1 glucose molecule. If you're with me to this point, you are doing such a great job. If you are confused, that is okay. Write down your questions. Watch the video again. Put in the time and it will have a payoff for you. Tomorrow in class, we will do all of this live. We will go over it again live and you'll be able to ask your questions in the comments. But I really need you guys to make sure that you're ready for class. Watch it again if you need to. Make sure you're prepared to learn about the Calvin cycle some more tomorrow.